to demonstrate how to use the environmental sampling kit that Membrane Protective Technologies supplies. First of all, you want to clean off the bench area or the tabletop that you're going to be putting all the supplies onto. So you can use a disinfectant, 70% ethanol, or isopropanol. So clean off the area. And then we will have the box. We'll open up the box. We take out all the supplies, which include the instructions, the PBS tubes, gloves, the blood auger plates, and sterile cotton swabs. So before we open any of the supplies up, we want to put our gloves on, and we want to try to keep sterile as possible. So at some point during this demonstration, I am going to stop talking because there's a lot of microbes and stuff in your mouth, and I don't want to contaminate with my contaminants the sample area. So once the gloves are on, then we will take everything out of the package. We'll take all the blood auger plates out of the package, and I'm just going to use this one right here. And then we will also open up the cotton swabs and feel for which end has the cotton part and which one is the end, the wooden end. And we'll open it up. And try to open it up to where you have the aluminum foil still on the one end. So then you can just lay it down on the bench top. And then we will also take the sterile PBS and the blood auger plates. We want to label the blood auger plates with the date and the location of where you're going to swab. So if you're going to swab your bench top or the water or the sink or whatever, you just want to have a location. So if you have contaminants, you know where to clean. So again, once I open up my tubes and everything, I'm going to stop talking, and then Bridget, my lovely assistant, will take over. So we have our cotton swabs. So we're going to take out a cotton swab or get a cotton swab ready. And then we're going to open up the tube.
And then you're going to dip the cotton swab into one of the PBS tubes, and do not put the wetted cotton swab down. Recap the PBS tube, and then swab the area of interest with the wet cotton swab. Open the lid of the blood auger plate labeled with that location, and swab all over the plate. Replace the lid of the blood auger plate when you are done.
So once you have done your swab for each location, so you'll do this at the various locations that you want. Once that is done, then you will take your plates and you will store them on the bench top. These are environmental samples, so they're growing at room temperature anyway, so they should grow. And you'll put them upside down. Okay, so you have the lid, you want to put them upside down. And we put them upside down because if you get condensation or something, again, you're going to have false contamination due to whatever's on there possibly. So you want that. And then let these sit on your bench top 24, 48 hours. It'll probably take about 48 hours to where it'll grow. Do not place these plates anywhere where you have a fan or ventilation or something going on them. The plates will dry out, but you'll also possibly get some contamination from anything that's circulating around. So it's best just to put it aside and forget about it for a couple of days. And that is how we do an environmental sample.